So good morning. My name is Brad Theilman, and I'm a postdoc at Sandia National Labs in New Mexico. I'm thankful for the opportunity to be back in San Diego to present some ideas that have been growing over a couple of CRCNS projects for this story that we're calling Decomposing Spiking Neural Networks with Graphical Neural Activity Threads. So there's a tension in neuroscience between the discrete and the continuous. This is something that has been brought up a couple of times already in this meeting. But on the continuous side, we know that dynamical systems theory has been highly successful over the last few years as a tool for systems neuroscience. The world at ecological scales is continuous, and the brain is sort of set up for producing continuous behaviors. And the way that this manifests in our analyses in neuroscience is that we take neural activity and we transform it into continuous values that we can then map to these high-dimensional spaces and look for structures like manifolds and other continuous structures and try to interpret those in the context of computations that the brain is performing. The one thing that I can't get over is that, at the end of the day, neurons produce discrete spikes, and we have plenty of examples where spike timing matters quite a bit. In this famous figure on the right, neurons can produce very sparse and very precise spike patterns in the right circumstances. So a question that I think is always worthwhile asking is it valid to reduce spikes in neural activity to continuous values? In my past work in the auditory system, it wasn't hard to run into situations where it was hard to justify a firing rate in the context of the operation of the circuit. And so that tension leads to the motivating question for this talk and this project, which is what are useful abstractions for spiking computation? Because this tension kind of informs how we think about spiking computation at a more abstract level. And this question also sits at the intersection between computational neuroscience, which is where I came from, and neuromorphic computing or brain-inspired computing, which is the work that I do now at Sandia, because we need these abstractions to be able to translate computational algorithms and ideas from the brain to these neuromorphic computers to hopefully impact the energy efficiency of AI and other situations. So I really like this quote from computer science about the purpose of abstraction. It goes, the purpose of abstracting is not to be vague, but to create a new semantic level in which one can be absolutely precise. So I want to spend a little bit of time talking about what abstractions are and where they come from. And this is going to be kind of out of left field, but I think it's fun for me to think about at least. But natural numbers are a useful abstraction. Where do they come from? How would I construct them? I can think of natural numbers as being equivalence classes, right? I can take the collection of all finite sets, and I can say any two of them are equivalent if I can find a bijection between them. And that takes all the finite sets, doesn't matter what the things are, and groups them into these equivalence classes, which I can label with whatever labels I want, but those labels will behave like the natural numbers. This is one way to define or construct the natural numbers, and it comes from an equivalence relation. And you can, iterate this and go deeper and build more abstractions out of the same idea. So you can construct the integers as equivalence classes of natural numbers, the rational numbers as equivalence classes of integers, and the real numbers, and so on and so forth. So I really like this story, and it outlines, I think, a goal to keep in mind about how to build appropriate abstractions for computation, which is that you have to define the appropriate computational units and then the appropriate equivalence relation on them. And from that, a new level of discourse can emerge to talk about a new level of abstraction. But there is an art to it, because it's very easy to define equivalences that are either too strong or too weak, right? If your definition of an equivalence is too strong, then everything is isolated. If it's too weak, everything collapses to be the same. And so the trick is to try to be somewhere in the middle, where you just have enough flexibility to allow all of the interesting structure to emerge, but not too rigid that you kind of lose the sort of unifying insight that abstractions provide. And I'd like to argue that current dimensionality reduction approaches actually make use of this logic, even though it's a little bit opaque. So in dimensionality reduction in neuroscience, what we often do is we take high dimensional neural data and project it to a low dimensional space. What we're doing, we can describe that equivalently as saying that these two high dimensional neural activity vectors are equivalent if their difference lies in the null space of some projection operator. And so while this is a little bit of a roundabout way to describe what dimensionality reduction does, I think it's a useful way to frame it, because it allows us to see where we are maybe making some assumptions that we could make different choices for. And in particular, one thing that I'd like to point out is a lot of these analyses are relying on the neural population activity vector, which is this idea that the relation that binds spikes into computational units is kind of like epsilon simultaneity with respect to a clock in the experimenter's rig, right? So these time bins are defined by external clocks, or clocks that are external to the animal. And when you think about it, there's no real reason the brain should respect this relation. So if this assumption is being baked into the abstractions that emerge from this kind of approach, then it might be worthwhile to think about, could we bind spikes with a different relation to get a different set of abstractions? And there's a good example of this actually from a study a couple of years ago, where if you look at raw neural data across trials with respect to clock time, you don't see a lot of structure in the data. But if you apply some statistical time warping techniques where you just realign time to a more intrinsic time scale for the circuit, you see a lot of structure emerge that wasn't readily apparent to the eye. So this was really interesting to me, and I wanted to think about: is there a way that we could take that insight and use it to kind of build a new point of view for thinking about how to group spikes together into computational units? So computation in the brain has to be supported by the synaptic relations between spikes, because the synapses are what mediate the interactions between spikes to propagate activity through the network. And so I'd like to propose that instead of binding spikes together into groups by time bins, we try to bind them by their synaptic relationship to each other. So this idea that if we could quantify the degree to which a presynaptic spike caused a postsynaptic spike, then maybe we could build groups of spikes that are sort of bound together by this relation and build new abstractions on top of that. And I think that this is useful because that notion of synaptic relatedness is intrinsic to the population and gets us away of this idea of gets us away from having to rely on the experimenter's clock to define the relation. So how might we do this? In the neuromorphic computers I work with, it's easy because I know every connection and I can do anything I want essentially. So what we did was we kind of came up with like the most naive quantitative measure that we thought captured the intuition behind the synaptic relation. So I'm not going to claim that this is the end of the story. It's just a beginning, and it's going to look very familiar. But we arrived at it from this point of view of trying to quantify this relation. So for any pair of spikes that occur at time beta or time alpha, I'm going to define this quantity that I call omega based on the synaptic connection between the neurons producing these spikes and the time of the spikes themselves. So it's going to be the product of the normalized weight between the neurons, a Heaviside step function that ensures causality. So we want to make sure that the postsynaptic spike is at least one synaptic or conduction delay after the presynaptic spike. And then this exponential decay factor, which is just supposed to, kind of build in the intuition that spikes that are separated in time should be less related to each other. So this is, clearly the postsynaptic response of a leaky integrating fire neuron. It's also, you could think of it as like the facilitation kernel for STDP. I think there are connections there, but I want to reinterpret these terms in this context of quantifying the causal relation between spikes. And causal is sort of a tricky word. I don't have a better one to use in this context. So, it's not meant to be related to statistical definitions of causality, just to capture this intuition of synaptic relatedness. So what I'm going to do is I'm going to look at a very simple test spiking neural network where I have 6,000 spiking neurons, Ajikevich model neurons, distributed on the surface of the sphere with a distance-dependent connectivity probability, so neurons connect to their geometrically nearest neighbors. I'm going to evolve it under random stimulation, Poisson stimulation, for about 10 minutes to let under standard STDP rules for the network to kind of equilibrate. And when you do this, this has been known for a while, and it's generally true that these kind of networks sort of settle into a sparse asynchronous firing regime where the individual neurons display Poisson-like statistics, and there's no obvious synchronization across the network. But now what I'm going to do is I'm going to compute that quantity omega between every pair of spikes. And I'm a pretty patient person, so that's a lot of computation, but it's actually not too bad. And then once I have that quantity omega, I'm going to apply a threshold to it. And if two spikes are related by a value omega that exceeds this threshold, I'm going to insert a directed edge between the presynaptic spike and the postsynaptic spike. And this is going to build a very large directed acyclic graph directly on the spike train itself and not the neurons itself. And this is kind of what that looks like. So you can start to see some fluctuations in the density of edges that hint at some structure. But one thing that we found that was kind of interesting was that if you look at the disjoint connected components of this large activity graph, we found that the activity sort of naturally split into these separated pieces or separated groups of spikes which I've indicated by different colors here. And we refer to these disjoint connected components as graphical neural activity threads, or NATs. The intuition is kind of, we were hoping to interpret these as sort of like a stack trace of the computation through the network, or like a computational thread. So how do we know that the NATs that I've defined are well defined? Because they depend on some parameters in our definition of synaptic relatedness. We could go into the details, but I think one thing that we did that was pretty convincing was that we computed the quantity omega for the empirical data as well as shuffled data, where we shuffle all the spike times to preserve the firing rate but break all the temporal correlations. And that allows us to get basically a null distribution for this, in this case, the negative log of omega. And we see that in the empirical data, we have an extremely large peak for large omega values, which to me indicates that the quantity omega is actually capturing some meaningful flow of information through synapses to the network. So we defined our threshold for omega by looking at this knee, where the peak sort of transitions back to the null distribution. And that's how we defined the edges that we included in the NATs. And if we change the parameters, this peak changes slightly, but not significantly. So that's why I would claim the NATs are well-defined and intrinsic to the population. But if we zoom into the NATs, I want to point out a few things that we noticed that we think are indicative of an interesting implications of looking at the neural activity this way. So what I'm showing here is a zoomed-in view of several NATs. And on top of that, I have 10 millisecond time bins superimposed. The first thing I want to point out is that within a time bin, spikes that belong to the same time bin could belong to separate disjoint NATs. And this is important because the fact that the spikes belong to different NATs mean their causal past and futures never intersect. So they never have, they're not read out, at least in this network, for any sort of future computational activity, even though the time bins would sort of bind them into the same computational unit. We can see that we can have multiple NATs operating in parallel, and we also see that the asynchronous. Well, sorry. Let me just move on to the next slide. So the way I wanted what I wanted to say was that the NATs, the structure that we get out of the NATs is kind of, you can think of it as like trying to unify the connectome and the spiking activity into a single mathematical structure, so that the NATs that we observe, we can project them back down to the connectome, but we sort of unfold the whole activity into this larger structure. And if we kind of spread out the network in time, and we look at if we flatten the network and look at it over time, we see these sort of like really intricate structures that emerge from the activity. So the question is, if these NATs are supposed to represent spiking computations, then what are these computations? That's a tough problem to address just without any sort of reference, but we might be able to make some progress if we weaken the problem by instead asking the question, when are two NATs performing the same computations? Because if we understand what the same computation means, then we can use this idea of partitioning NATs into equivalence classes to define the computations. So we need a definition of NAT isomorphisms. And the ideal candidate would be to look at graph isomorphisms. And that's a computationally hard problem in general, but this isn't the general case. What we can do is, if we imagine we have two NATs that are isomorphic, we can kind of take their Cartesian product, and so connected components of this Cartesian product would pull back to the isomorphic threads that are shared in this isomorphism. So a connected component of the Cartesian product would indicate that we have two NATs that have the same causal structure. And we can actually do this computation relatively efficiently by using the right data structure. So I won't go into detail on how this computation works, but suffice to say, we can use this to efficiently extract isomorphic NATs from large spiking data sets. And this is an example of what one of those looks like. So on the top, I have a set of spikes that are connected by some relations, and on the bottom, I have spikes that occurred widely separated in time, but still share the same structure of the synaptic interactions between them. And importantly, the spike timing is not preserved. And you can see that in this example over here. So we can extract isomorphic NATs of a wide variety of sizes, but we can also see that this definition of isomorphism sort of builds in the flexibility that the same patterns can recur with flexible timing relationships, which is something that wouldn't be captured if you were restricted to a time bin approach. So what I'm going to do to kind of get closer to the idea of trying to understand what the NATs might be computing is I'm going to pick a fixed input pattern of spikes. I'm just going to apply this to this network over and over again in different trials. And what I noticed was that these input spikes will trigger gnats to emerge in the activity. But within those gnats that are triggered, you might have specific isomorphic subthreads that are shared between the gnats that are evoked on different trials. So in this example on the left, I have this red gnat that is evoked, and it has this subpattern that's given in black. And then later on, when that input is represented, a different gnat emerges, but the same subpattern is shared. And so now we can build another graph where the vertices are now the individual gnats or disjoint components that we extract from the data, and we can connect them if they share a subgnat. And this is a multigraph in general because different gnats can share multiple subgraphs. And when I do that, what I find is that the gnats that are produced by the network kind of naturally decompose into these really well-defined clusters, where again, the black edges indicate shared patterns of activity between the threads. And when I pull those clusters back to the actual spike rasters, what I see is that on individual trials, the spiking response with respect to the time bins is actually very different. But what the gnat analysis gives us is a way to say that in certain intervals of time, the evoked activity embedded in the larger spiking activity shares some sort of isomorphic structure that isn't apparent just by the temporal relations between the spikes, but between the synaptic relations between the spikes. And they occur in a very repeatable order. But again, the timings and there's a lot of flexibility in what happens on individual trials. So this gives us a picture where we have, a spiking neural network that's sort of generating its own activity, generating these own threads, where activity is, being propagated into, influencing the threads and maybe coming back out. And in this CRCNS project that we're starting, I'm trying to connect this to dynamical systems by using linear readouts to sort of map spikes to dynamical modes in a sort of behavioral system, and then use dynamic mode decomposition to kind of build a quantitative measure of how sensory and motor spikes interact through the dynamical system. So with this quantitative measure, I now potentially have a way to extend the directed graph of the gnats through a dynamical system to complete this sort of picture of a sensory motor loop with respect to this idea of activity propagating through synapses. And then I unfortunately don't have time to go into details about the mathematical basis to this, but there's some interesting connections to topology that I think will be interesting to explore. And also at Sandia, because we have these large neuromorphic computers that are very efficient at simulating biological connectomes, such as Felix presented our work on simulating the fruit fly connectome on a neuromorphic system, we can now do high throughput experiments extracting gnats from actual biological networks. So with that, I'd like to thank the CRCNS program for funding this work over a couple of projects. And then the code for extracting these gnats is available on GitHub. And we also have a preprint describing some of the methods available here. So thanks for your attention.
So experimentalists typically have access only to a subset of the neurons of the big network.
Yeah.
Like the one you presented. So
yeah.
I was wondering if you try to apply your technique to only a subset of these 200 neurons, and to what extent you can recover the gnats.
Right. That's a fantastic question. What I'd like to say is that, like in this example right here, if I have a larger network and I cut out a piece of it, there's going to be some gnats that exist within that piece, and those are going to be embedded in this larger gnat, which we don't have access to. But within that, we can extract the sort of patterns of computation that are available within just that subnetwork, and then kind of use this boundary to give us like channels for interaction, being like whatever gnats happen in this subset have to interface with the rest of the network in these particular ways. So it's a longer story, but that's how I'd go about answering it.
Sounds like a natural thing.
Very good.
So did I understand that correctly, that gnats can operate at different time scales?
Correct.
Can you give us an intuition of how?
Well, so I mean,
Can you repeat the question?
Yeah, sorry. So the question was, gnats can operate at different time scales. Can I give some intuition about why that might be the case? So the neurons are embedded in this larger population where they are receiving spikes that may be weak, or the timing might be right, so that they don't directly influence the postsynaptic spiking, but they do modulate the state of that neuron. And that can alter how that neuron responds to, like a stronger input that actually does propagate spikes. And so through that mechanism, I think that, the timings of the individual spikes within a gnat can be modulated.
Okay.